Hi, my name is David. Today I'm going to be going over the ACT Science section for test 403. So there are 40 questions, so let's get to it. All right. So passage one involves metabolic rate and basically the energy consumption of the animals. We have a graphic right here. We got a table of values down there. All right. But whenever you're doing an ACT test, you definitely want to look at the question first. So let's look at the question. According to figure one, an organism with mass 1 gram would have an MR closest to blank. So figure one's right here. We want to look at mass, which is here. Again, it doesn't say 1, but we want 10 to the 0. And so we got a number right there, which looks like 10 to the negative 3. So the answer is A. Question number 2. Based on the information in figure 1, if a Komodo dragon, a reptile, and a sheep mammal, so again, cold-blooded organisms, poikilotherms, warm-blooded homeotherms, had equal weights, which of the following would be their MRs? So again, we don't actually have beats per minute here in this figure 1. You may be tempted to look down at the table, but it says figure 1, so stay focused on the figure. One thing you can notice is that the poikilotherms, the cold-blooded reptiles, are underneath the warm-blooded. So that's why it could be any of the numbers where the first one is less than the second one. And since only A has that, A is the answer. This time we're looking at the table. In the table, the giant Galapagos turtle has an average heartbeat of 9 beats per minute, which could be following could be the lifespan. So it's not on the table, but what you could kind of imagine is that as the heart rate goes down, the lifespan goes up. So it should definitely be more than 75. Now the question is, it looks like when it went from 30 to 18, it kind of subtracted 12, but went up by 30. Or more likely, it divided by 2 and then it's like times by 2 approximately. And you can confirm that by comparing better numbers like 90 to 30 and 15 to 45. So this divided by 3 times by 3. So because because 9 is half of 18, what we're going to want to do is double the 75. So we get 150 is our answer. So definitely kind of tricky, but you might imagine that no tortoises do live for a long time, but they don't live to be 6,000 years old. So, all right, that was C. Question number 4. Which of the following statements best explains the trend in figure 1? Well, I notice as mass goes up, the metabolic rate increases. Large organisms consume more energy dense food. That does not seem to be listed on here. It doesn't talk about any about the food consumption. Larger organisms are more likely to be carnivores. That's true, but the woolly mammoth at the end is a herbivore, so it's not true. Large organisms are exclusively mammals. Nope, we have the tortoise right here. Larger organisms require more energy to survive. That's true. As they get bigger, they require a higher rate. Question number 5. Which of the following best explains why heart rate is used as an indicator of MR? MR, metabolic rate, how fast your metabolism is going. Heart rate beats blood, so that kind of makes sense that's connected. You could have also done like breathing rate too. Heart rate remains constant regardless of activity center. Well, we would not want that because of the fact that we would want to see how the metabolic rate changes. B, the heart supplies nutrients and oxygen, so a fast heart rate suggests a higher energy use. That sounds relevant to our question. Heart rate only increases with age, not metabolism. Well, we don't have anything about age. We have mass on here, so we can eliminate C. And heart rate is only affected by body mass. I mean, it could be related to it, but I think stress could be a case that could affect your heart rate, such as like seeing a lion jump into a room might make me get super scared. But the B is the best answer to this case, and that's unfortunately sometimes with the ACT, you have to pick the best of the 4 choices, not actually have a very good solid answer. All right, there are 2 more questions for this passage. Which organelle is most directly responsible for the metabolic energy used by the eukaryotic organisms? Well, as you might guess, it's the mitochondria, which is known as the powerhouse of the cell. So I hope you got that one right. But if not, maybe that'll help you remind yourself that mitochondria helps break down glucose and gives the cell energy for eukaryotic organisms, like plants and animal cells. Last one, the relationship between variables in table 1 is best modeled by which of the following equations? All right, so is heart rate divided by 150 lifespan? So checking that. Heart rate divided by 150 is 3. That does check. 150 divided by 150 is 1, not 9. So it's not A. Mass times heart rate is 150. 0.06 times 450. I don't know. That's hard math. 1 times 150 is good. 5 times 90. Nope, that's 450. So it's not B. All right, lifespan times heart rate is 1350. Main, I guess it looks. Let's just check D. Maybe that's easier. Heart rate times mass is lifespan. Heart rate times mass. So that was, we checked that already. So that's not always, so that right there would break on the first one because it's supposed to be 150, not 9. So that's not right. So the only one that was correct was C. So you don't have a calculator for this test. So definitely sometimes you want to go through the different choices as that might be faster. And you definitely can estimate a lot for doing the math here. Passage 2 involves two figures, table 1 and table 2, and involves entangled photons. So first off, some little background knowledge in case you were not aware. A photon is a packet of light energy. You may have gotten confused with something called a proton, which is a subatomic particle found in the nucleus. All right, but let's go through it. So in course, the first question, table 1, which of flying trials did the scientists change P2's initial state? So we looked at table 1, we have P2's initial state. So this one, it stayed the same. So not trial 1. Trials 1 and 3 will not trial 1. Trial 2 and 4. So that one stayed the same. So it has to be D. Let's verify. Plus changed to minus, minus changed to plus. All right, so the answer is D. Based on the information provided in the passage, which flying would most likely be considered a classical system. So if you don't know what a classical system is, they talk about that here in the first sentence. In classical physics, every object have mass are considered independent, where changes of an object's properties do not affect the properties of a different object. So the idea of if this table that I'm working at went from purple to green, it would not cause other things on the table to change. It would make the keyboard change color, which is what most normal things that you think about follow. All right, so what things might be like that? Well, I don't know how quarks work and then make a proton. I don't know how photons in a lamp work. I don't really know how electrons work, but books on a shelf, that sounds like everyday objects. So definitely physics gets kind of weird as you go further down to subatomic particles, which is kind of cool. But in this case, you're supposed to pick for the simplest answer, the most logical conclusion, instead of like reaching for it. What can be concluded about the states of entangled photons in table 2? Well, let's look at them in table 2. And one thing I can guess EP is entangled photons. It also mentions that in here. The entangled photons have the same states. Well, one thing I noticed right away is they have opposite states. Oh, so that looks like C, but we can check B and D to make sure. The state of one entangled photon changed while the other did not change. Well, it looks like they both change. In order for both states to change, the scientists had to modify both of them. Okay, well, that could be true. Let's read the directions. In quantum physics, particles can become entangled, so the actions from one affect the other, even when they're very great distances. In order to achieve this, the scientists used a laser and split a photon into 2 entangled photons and measured the initial final states after she modified one of the photons. So she did not modify both. She only modified one of them, but then they both changed. So the answer was C. Scientists want to test the speed of information transfer between 2 EPs, or entangled photons. One EP was transported to the moon, so we got the moon here, and probably the other one's on Earth. So we have EP1, and let's just say EP2. The other remained on Earth. A change is made to the EP on Earth, like what happened in this trial. So one of them changed. And so it could have been this was +1, this was -1, but then they changed it to -1, and then that would change to +1. If information between 2 EPs travel 10 times faster than the speed of light, and a radio signal takes approximately 1 second. Okay, that's kind of weird that they say radio signal. But now, as you might guess, a radio signal is a form of light. So that means a radio signal would take 1 second to go between Earth and the moon, and an EP would take, it goes 10 times faster, so an EP would take 0.1. So let's just write some notes down. Radio is 1 second, and then EP is going to be 1/10 of a second. All right. How long would scientists on Earth have to wait to receive a radio signal from the moon-based detector? Okay, so they change on Earth. That then takes 0.1 seconds to modify the EP on the moon. Then the sensor on the moon detects it, sends a radio signal back, which will take 1 second. So then the total time looks like it'll be 1.1 second, which is going to be B. And I apologize for the messiness of the handwriting, but hopefully that was a little bit easy to read. So again, we had 1 second for the radio wave, 1/10 of a second, the EP. And again, the key thing was that you had to wait for the EP to change and then wait for the radio signal to go back. So if you did 0.2, it was like EP there, EP back. That might make you think of A. Or if you were only thinking about the EP, the time it takes for the Earth to the moon, that might make you think of A. If you're thinking radio wave there and back, that would be 2 seconds, one radio wave, so 1 second there, 1 second back. Again, the goal here is that we did not have to send a radio signal to the detector saying we changed it. The EP changed, then the moon detected it and then sent it back. So tricky question, but hopefully that made a little bit more sense. All right, trials 1 and 5 differ in what respect? So this is comparing 2 different experiments, but we have trial 1 and trial 5. So a couple things I noticed is that this +1 and this is a -1, and this is a plus. Oh, wait, that's the only thing that's different besides those two. Another thing I noticed difference, it says EP versus P. In trial 1, one of the photon states changed, while in 2, both photon states changed. I mean, that's true, right? We had only one change from plus to minus. The P1 changed to plus to minus. In this case, both changed. In trial 1, both changed. Nope. In trial 1, photons were used. In trial 5, electrons were used. Nope. The E was not electrons. It was entangled photons. So C and D are out, and the answer was A. All right. Next, the particle used in table 1 and 2 are massless. Okay, perfect. Does information in the results of table 1 and 2 support the hypothesis that massive particles are always entangled? Well, I'd say no, because this was not entangled, and this was entangled. And they're both using photons. It says that they're both massless. So the answer is not yes, it should be no, because the particles are independent in table 1, but not in table 2. That sounds good. The particles had mass. No, they were massless. So again, sometimes the ACT has weird contradictory information, but again, there's only one correct answer, so they have to try to trick you somehow. So the answer was C. And that was the last one for passage 2. On to passage 3. So passage 3 has a lot more information. Looks like a chemistry setup. We have some experimental design, which definitely gets asked about on the ACT. However, I don't usually read all this because of the fact that it takes up time, and then I only need to use this if it asks me about the experimental design setup. We have some PPAAs, essays. So real quickly, I noticed that PP is propanol, which is whatever that is. Acetic acid is AA. Sulfuric acid is SA, which is a catalyst. So that seems kind of important. Okay, and then PA is something else. Oh, or there you go, propyl acetate. Sure. And then it looks like we did 3 experiments. All right, anyways. Based on the results of experiment 1 and 3, to remove the drying tube did what? So we want to compare this to this. Looking quickly at experiment 3, it says 4 and 6 of experiment 1 were repeated, except the drying tube was not attached. So we typically the independent variables are at the left, and then the dependent variable, the thing we're measuring, is on the right. So it looks like we're looking for trial 17, 18, 19, and comparing that to 4, 5, and 6. So we have 1, 2, 1. If we go to 4, 5, 6, we have 5, 10, 10. So it seems like the PA was way less for trials 17 to 19. So the remove the drying tube reduced the amount of PA. So B is the answer. The prime purpose behind the addition of SA to the reaction was to increase what? Well, first off, SA, it said, was a catalyst. So usually with catalyst, it shortens reaction time. So it's not C, it doesn't increase reaction time, it doesn't increase temperature. It definitely doesn't increase reactance. So generally speaking, it should make the reaction go faster. Now, in a way, that makes A be the correct answer, because in an amount of time, you would get more product in the amount of time. So it's definitely something you may have originally crossed off. And if there was something that said it said decrease the time, then definitely I'd pick that answer over that. Or if it also said lower the activation energy, that's something I would pick too. But in this case, the best answer is picking the amount of product. Question 16. In a chemical reaction, the limiting reactant is the reactant that runs out first and limits the amount of product that can be produced. In which of the following trials was AA the limiting reactant? So limiting reactants, by the way, must always be on the left side of the chemical reaction. Reactions are always on the left, products are on the right. So let's look at trial 2. So for trial 2, we kept AA the 5 as we kind of go down the list, but as PP increased, the amount of PA increased. So that tells me that AA is not the reactant for trial 2. How about trial 6? In trial 6, we have PP go from 10 to 20, and we kept AA the same, but the amount of PA did not increase. So that tells me that trial 6 must be the limiting reactant because of the fact that we increased one of the reactants, but we didn't get any more product. Question 17. Suppose another trial was done for experiment 3, but this time 30 milliliters of PP was introduced. How much PA would be produced? Well, we did 5, 10, 20, 10, 10, 10. But one thing we talked about earlier is that the drying tube removed out kind of reduced the amount of PA produced. So how much should we expect? Well, I would not expect a lot, especially compared to what we may have seen on here, where it's like if it was 20, 10, and 4, we got 20. So the answer is D, because we would expect the PA to be similar to what we had earlier, because increasing the amount of PP does not always increase the amount of PA, right? Well, it's increasing from 1 to 1, so it sounds like it should actually probably be around 1 or 2, and that's the best answer. A student in another chemistry class tried to replicate trial 3, but used calcium sulfate instead of calcium chloride. Yeah, well, what were those used in this case? So checking out there, it says calcium chloride was a drying agent, and that removed the water byproduct and to react with PA and other organic compounds. If 2 milliliters of PA was collected, does this result suggest that CaSO4 is better or more effective than CaCl? Well, let's compare it. So we did trial 3. Well, normally we get 4. In this case, we got 2. So it sounds like it's worse. So it's not A or B. So cross those off. No, because the amount of KX is greater. No, it was less. No, because the amount of KX is less than the amount produced in trial 3. And that's why the answer is D. Question number 19. Which of the following best explains the results of trial 7 and 8? So trial 7 and 8, let's compare them. Well, we had PP go down, AA go up, but we had the sulfuric acid stay the same. So it's hard to compare these two because both of them changed. So what we may want to do instead is try to compare something else, or maybe like we can see like what would happen if we compared 10, 10, 4 or something like that. Unfortunately, we don't have a good option here. So let's go through the choices that we have. As SA increases, PA decreases. Well, as an overall rule, that doesn't seem true. It seems as when SA was low, PA was low and SA decreased. So that seemed wrong. But also, SA did not change from between these two. So as time changed, well, that was involving trials 9 through 16 is when we changed the time. So we can ignore B. As PP decreases, PA decreases. Well, when PP went down, PA went up. So that PA went down. So that seems right. As calcium chloride decreased, well, we didn't actually change the amount of calcium chloride. So that was constant for the experiments. So we can eliminate D as well. Which of the following statements gives the most likely reason the vial was heated to 175 degrees Celsius as shown in figure 1? Again, most likely reason. So it probably didn't tell us how. But that's going back to the experiment design, where they were mixed in a conical vial. A drying tube was attached to a condenser. The vial was attached to liquid mixture to the condenser, and sandbox was up to 175. So one thing you might imagine is you put something that hot. Again, at 100 degrees Celsius, water boils. So it's kind of like at 175, the water should, any of the liquid should be evaporating. So that's kind of what our goal is. We're not trying to condense the material. We're trying to evaporate it. And we're not trying to evaporate the gases. We're trying to evaporate the liquid reactants. All right, that does it for passage 3. On to passage 4. Based on this passage, which involves R selecting case-selected species from biology, We also have a survivorship curve, which has type 1, type 2, type 3. All right, let's go to the questions. Based on the information provided by the figures in the table, which of the following is most likely a type 2 organism? Well, as says here in figure 2, there's K selected species, songbird, and humans, over 1 R selected frogs. I feel like we're supposed to just guess based on this question, type 2 organism. We also have this table here. So we're probably supposed to make a guess at this. So let's look at the creatures. Well, we have oysters here. Oysters, they die very rapidly. And that's probably going to be a type 3 because the percent of organisms surviving is very low. Most oysters don't live to their maximum lifespan, according to this table. How about squirrels? Well, squirrels have a steady drop. It's like 25, 20, 25, 25, 25, which is like songbirds here. How about sunflowers? Sunflowers look similar to oysters, where it's kind of like it decays rapidly. And then sequoias, it takes a very long time. So sequoias look like type 1. So the best answer here is squirrels, and that's because type 2 should go down at a constant rate, and that's what we see here as table 1, 2. According to figure 1, when a species approaches the carrying capacity, the population tends to do what? So looking at this, it seems like it goes up and down around it. The K selected one is more calm, and then the R selected is more rapid. It does not increase indefinitely, does not decrease back to 0. It does not remain constant. The word oscillate. Oscillate means it goes up and down. So that's why D is the correct answer. Based on figure 2, which will find correctly ranks the species by age at which 75% of the population is still alive. So 75% of the population is still alive. That's probably roughly here. So all right, so which ranks the species by age? So definitely the age of type 3 is the youngest. So type 3 should be the smallest. So they put the inequalities backwards, but definitely A or B now. And then type 2 is next, so then the answer is A. So watch out for those inequality signs. They sometimes go that way, sometimes go that way, so make sure to check it. In the event of secondary succession, so that's a term you might have seen in AP biology class or AP environmental sciences, the ecological recovery of the area after a disturbance such as a fire, flood, or human activity. Which type of species would best suited to repopulate the area quickly? Well, R selected species reproduce rapidly. So probably any R selected would be better at replacing anywhere. So reproduce rapidly, may have many offspring, that's true. They don't necessarily thrive on stable environments and grow quickly, but they definitely reproduce rapidly, which is in the passage. And then these two are not true because K selected is slower growth. An increase in the amount of available food in land would have what effect on K selected species? Well, the whole point of these species is that they go to the carrying capacity. If you increase the amount of resources available, the carrying capacity increases. So it would probably cause a gradual increase and then the whole oscillating or leveling around that new level. Approximately 5% of clownfish are hatched from a given clutch of eggs are still alive 3 years later. If clownfish typically live for 4 years, so that's about, it looks like they hit the lifespan of 75%, but only 5% of them remain. So here's 75%. Okay, that's pretty low. That's relative. That's like about 25%. That's much higher, like probably 75. So I'm guessing, based on these numbers, that this clownfish is probably a type 3. And that is one of the options. I don't know if it's K selected or R selected, but we definitely can tell it's type 3. Okay, now that does it for the fourth passage. On to the fifth passage. So the fifth passage is definitely very intimidating with the big graphic here. Again, there's a lot going on, but it will make sense as we do more of it. There's also 3 experiments too to watch out for. So let's get to it. So if a similar setup to experiment 2 was performed, so we want to look at how this experiment was set up. With the gap size decreased by a factor of 2, which of the following cells would not be bumped? Okay, so I don't know what's going on with those terms. So we could look here and say the gap size decreased, but I don't know if decreasing the gap size will make it be more bumped or less bumped. So I could skip this question and come back to it, or I could spend some time reading about what the words are. So an introduction of the material. a bump array that channels fluids to sort particles by size. Okay. And then they talk about the word bumping to one side, while those of smaller threshold continue to flow with the fluid. So it seems like this small particle goes with the fluid flow, but these bigger ones go along the grid. So they're forced. Okay. Well, I mean, which line would not be bumped? I'd probably pick the smallest one, just by the bare minimum. Here, if the gap cell size is 5 and was not bumped, if you decrease the gap between them, make the poles closer together, that seems it's more likely to be bumped. But comparing 5 versus 1, I'd pick 1. So it's kind of an interesting question, but definitely pick the smallest. Using the information in the tables, which experiment would be most accurate in separating cancer cells from normal cells? Well, I don't know the size of cancer cells, but looking at this table 1 here, we do see that white blood cells are 15 and cancer cells are 20. So we want something that does not bump both. It doesn't bump either, but bumps the cancer cells that are bigger from the white blood cells. Let's see what does that. Looking at experiment 1, 15 is not bumped, but 20 is bumped. So experiment 1 is good. And there's only one with experiment 1, so that's pretty good. We check experiment 2. Experiment 2, it bumps at 10, so that'd bump both of them. So it's not B or D, and then C. Experiment 3, which is table 4. But yeah, that also bumps at 10. So yeah, experiment 1. A scientist is thinking of using a large-scale bump array to filter cancer cells from the patient's blood. How many cancers would they expect to extract from 6 liters? Okay, so we have 6 liters, and then this right here for cancer cells is 10 per milliliter. So there are 1,000 milliliters in 1 liter. So you can think of a millennial is the year 2000, there are 1,000 years. Millennium is 1,000 years in a year. So 1,000 milliliters in a liter. And then we multiply that by, it looks like 10 cells per milliliter. So that would be, whoops, I have these backwards. Let's go back a little bit. 1,000 milliliters on top, liters on the bottom. That way the liters cancel. And then 6 cancer cells for every 1 milliliter. All right, I don't have a calculator, but 10 cancer cells for 1 milliliter. But 6 times 1,000 times 10 is just going to be 60,000, which is not 60. It's not 6 billion. It's going to be B, or 6 times 10 to the 4. Which of the following was kept constant throughout the 3 experiments? So one thing I can see pretty quickly is that this one, it had the radius was used, so it's not radius. The next one was they did a different shape, so it's not shape. The gap between the posts. This had gaps of 30 versus gaps of 60, so it's not gap. Spacing between the center posts or the results of cells are bumped. Well, the results were changing, so it's not that either. So the spacing between the center of the post must have been constant. All right. Question 31. Suppose that breast cancer tumor cells are only slightly smaller than normal cancer cells at 14 micrometers. So again, that's also smaller than the white blood cells too. Given the information, if a 10 milliliter blood sample is used in a circular post-op with gaps of 60, would these cells be bumped? So how can we figure that out? We have to find the experiment that did that. So we can't look at experiment 2 because the gaps were 30. We can't look at experiment 3 because those were triangles. So we probably want to look at experiment 1. So it said circular post, radius for 5, the distance between the posts was 70, leading to gaps of 60. So this is basically telling you to look at experiment 1. Well, at 15, it was not bumped. So 14, since it's under 15, will also not be bumped. So the answer is no. No, because they're smaller than 15, which were not bumped, or no, because they're smaller than 20, which were not bumped. Well, the 20 is not true, and C is definitely true. And that's why C is the answer. What was the effect of swapping out circular-shaped posts for triangle-shaped posts? So experiment 3 was the same as experiment 1, but they did triangles instead of circulars. And what we noticed is it has the same results as experiment 2, which had bigger radii. So what I imagine is that the triangle had weird shapes or made it seem kind of bigger, which also reduced the threshold. So not the number of cells, but it's not the threshold size was increased, but the threshold size was decreased. So that way it caused the the cells had to be smaller for it to not get bumped. All right, the last one for passage 5. What could be the new threshold size if experiment 2 is repeated using triangular post? Well, I would imagine since experiment 3 had similar results to experiment 1 with triangle, making it have a bigger triangle would probably make it be further bumped. So I'd probably guess 5 or less. I would definitely not guess it would get less. The threshold size would get more like the first one because the triangle posts got bigger or seemed to block them more. So, but yes, 5 is there, so I'd probably pick that as the best answer. All right, on to passage 6. All right, passage 6 is our conflicting viewpoints. We always have one of these per test. And in this case, we're comparing 2 different scientists. So let's take a moment and gather what they're talking about. So generally speaking, you want to kind of read the blurb to kind of understand the argument. So one thing I see right away is that they're trying to explain the purpose of evolutionary yawning and the phenomenon of contagious yawning. So how can we go about this? Well, first thing I notice is that the yawning of scientist 1 thinks it's a physiological, where scientist 2 thinks it's a social response. Okay. Now, generally speaking, we have the 2 points. Now, they usually do like supporting facts to support their claim. So we have this one where it has the evolution of jawed vertebrates. And they use it to regulate their body systems. So it's like temperature and alertness. Sleep-deprived individuals tended not to yawn when they had cold compress. So it's something you put to their forehead. Furthermore, individuals who rate themselves of having enough sleep tend to yawn more when placed in a warmer environment than a cooler environment. Since yawning seems to be based on body temperature surroundings, contagious yawning is probably a result of individuals in a similar state. Okay, cool. So which scientist would assert that yawning is linked to sound of vertebrae? So definitely scientist 1, but how about scientist 2? Yawning is probably a vertebrate social response. Perfect, both of them. Now, if I click on the next one, the notes I just made went away. So something you may want to do is make your comments on one of them and go back and forth. You can also highlight anything on the new passage too. Which scientists, if any, would assert that external factors influence how likely humans are to yawn? Well, scientist 1 said about temperature, so definitely scientist 1. And scientist 2 said when meeting a friend, waking up. So it sounds like that is also an external factor. So again, it would be both scientists 1 and 2. Recent evidence show that chimpanzees who are closely related to humans, okay, they're also vertebrates, which is why we can apply this study, yawn at regular intervals regardless of the environmental temperature. So that sounds like it's going to weaken scientist 1, who is all about the temperature. Oh, and that's the question, scientist 1. Now, does it weaken scientist 2? Let's find out. It's a nonverbal communication. Again, they're saying it's not physiological. So, and it's all based on in there. So this does not say chimpanzees yawn at regular temperatures, whether they're seeing other chimpanzees yawn. So in this case, it's going to weaken scientist 1 primarily. Which of the following is most likely type cell mentioned by cell type scientist 2? Well, it was not in the beginning. So we have breathing here. Dogs tend to yawn. Oh, brain mirror cells. So brain is made up of not adipose white cells, white blood, or muscle cells. It's made up of neurons. So that's an outside knowledge question. A different study measured the sleeping breathing rate of humans with air temperatures 20 degrees Celsius and at 25. So basically cooler and hotter. So it seems relative to scientist 1. The studies had a 20% higher breathing rate at a higher temperature. Okay, so kind of goes on the yawn more in a warmer environment than a cooler environment. Scientists 1 responded to finding by stating that what? There's more oxygen in the room? Well, no, scientist 1 didn't talk about oxygen at all. So we can eliminate A and B. The warmer air is more effective at cooling the air. Well, if it was better at cooling, then people would yawn less at warmer temperatures and not yawn less at cooler temperatures. So what you might have been looking at is cooler air is more effective, but then the opposite of that is warmer air is less effective. How does scientist 1 respond to scientist 2 claim that people yawn when they see each other? Well, he's all about temperature. So A says mimic facial expressions. That's scientist 2. People in the same room yawn because they're experiencing the same temperature. That sounds perfect because temperature related. Is coincidence that people yawn as seeing each other? No, they're talking about temperature. And people are ill and have caught a contagious yawn. Not quite the same contagion that they're thinking about. This is more about you seeing something or feeling something doing it, not actually catching a bacteria or a virus. So B is the correct answer. All right, the final question for this passage. A study tests contagious yawning in 3 groups: 18-month-old, 3-year-olds, and adults diagnosed with Parkinson's disease. Scientist 2 claims that people would display contagious yawning. All right, well, let's see. Scientist 2 said that does not occur in infant groups younger than 2 years old, so definitely not 18-month-olds. So not A, not maybe D, or people with Parkinson's. Oh, well, so neither A or D or C. So the answer is B. Now, it did say younger than 2, so you might have been tripped up by that because it says the number 3 here. But because the goal was that it's younger than 2 or people with Parkinson's, so the 3-year-olds are in between that range, so then they should be showing it when they see it. All right. Hope you found that helpful, but definitely feel free to go back and review the information, try highlighting the information in the text, making notes. Yeah, thank you again.